Okay, I'm going to start by talking about mutagenesis, and it is induced mutations, which is environmental factors, which is your mutagens, and that causes a mutation and can be imposed by the researcher. Mutagens can be chemical, physical, or biological agents that induce the mutations. Chemical mutagens, think of like the Green Goblin that's in the Spider-Man movie, and the nucleotide base analogs. The definition of this is resembles nucleotide bases but have faulty base pairing. So the DNA polymerase cannot differentiate and grabs the analog instead of its actual real base. Aminopurine is substituted for adenine, which results in your wrong base pairing. And a result could be a mismatched base, like a single base substitution. And then chemicals can cause modifications to A, T, or G, or C. Example is your alkylizing agents. They introduce changes in their replicating and non-replicating DNA, results in the incorrect base pairing, the single base substitution, like I talked about before, and then chemical mutagens that cause frame shift mutations as well, just not substitutions. An example of that is intercalating agents, and they just insert themselves between two bases, pushing them apart and triggering insertions or deletions. And those are type of mutagens. And to keep going on, now we're going to talk about the physical mutagens, which radiant and radiation, and you can think of any superhero movie that involves some sort of radiation due to a broken particle accelerator, and it creates stronger mutations, like mutants, than it had before. And so there's non-ionizing radiation and ionizing radiation, and I'll first talk about the non-ionizing radiation, which an example of that is just your UV rays, your ultraviolet. It has a lower frequency. Purines and pyrimidines strongly absorb the UV. Primarily forms pyrimidine dimers, which is two adjacent C's or T's on the same strands that become covalently bonded. It kind of like kinks the DNA structure, and the DNA polymerase has a hard time doing its job once that happens. And killing cells by UV is primarily due to the effect on the DNA. And then now we'll talk about the ionizing radiation. An example of that is X-rays, cosmic rays, and gamma rays. That is a higher frequency and it's more powerful than the UV. It ionizes water and other substances, results in free radicals such as hydroxyl radicals, which is your OH minus, and that damages your macromolecules. And it causes double and single stranded breaks and can lead to rearrangement to large deletions. And then to continue on with mutagenesis, we're going to talk about DNA repair and the SOS system. So mutations are heritable. Hereditary. They're hereditary. You can get them from somebody. So if damage can be corrected before cell division, and then you'd have no mutation occur. So like in bacteria, the stalled replication or major DNA damage activates the SOS repair system, which initiates many DNA repair processes, some which are error-free. It allows the DNA repair to occur without a template. Example, like with a random incorporation of DNTPs, and resulting in many errors and mutations. So in E. coli, the SOS controls transcription of approximately 40 genes. And the regulators are your Lex A, which is your repressor, and your Rec A, which is normally for recombination proteins. And so now I'm going to talk about genetic recombination. And first, we're going to talk about the horizontal gene transfer, which is the gene movement between cells that are not direct descendants. It allows for quick acquisition of new characteristic, fuels metabolic diversity, and there's 3 different mechanisms of bacterial genetic exchange. And that is transformation, transduction, and conjugation. And the 3 fates of it is degradation, replication by itself, and recombination within the host genome. So, recombination is a physical exchange of DNA between the genetic elements, which is your structure of your DNA. And homologous recombination is the process that results in genetic exchange between different DNAs. So, like from 2 different sources, it drives crossing over in classical genetics, which is during meiosis, and the REC-A protein helps recombination to occur. So, some effects that recombination has on your genotype is the sequence must be related but distinct. So, example, like your diploid eukaryotic cells with chromosomes from each parent. In bacteria, genetic recombination occurs after fragments are transferred by transformation, transduction, or conjugation. And to detect exchange of DNA, the recombinant cells most phenotypically differ from both of your parent cells. And then in lab experiments, we usually use recipient strands lacking a selectable characteristic that recombinants gain. And then complementation occurs if a functional wild type copy is supplied on a plasmid or bacteriophage, and it restores the wild type phenotype. And so now I'm going to talk about transformation. And transformation is the genetic transfer process by which free DNA is incorporated into the recipient cell and brings about genetic change, which could be cell lysis when that happens. Typically, your transformable cell is about 10 genes and 10,000 nucleotides, so only a small portion of the gene is transferred in one event. Competence in transformation is you're competent as an acceptor cell that can take up DNA and can be transformed. It can be induced in the lab. In some bacteria, competence directly is linked to your pili or your secretion system protein. And there's 3 stages of transformation, which is binding of DS foreign bacteria to your pili or your secretion system, uptake of DNA, which is your degradation I cannot say that word, of complementary DNA strands, and then REC-A is mediated to your recombination of the new strand into the bacterial genome if the foreign DNA is homologous to the host DNA. And then the recipient cell is considered to be transformed. The plasmid DNA must remain DS and circular for replication. And then I'm going to go and talk about my last topic, which is transduction. And transduction is the transfer of DNA from one cell to another by a bacteriophage. And there's 2 modes of transduction, which is your generalized transduction or your specialized transduction. The generalized is when DNA from any portion of the host genome is packaged inside the viron. The donor genes cannot replicate independently. They will be lost without recombination, and sometimes host DNA accidentally is packaged into the phage, forming transducing particles that is defective and cannot lead to viral lytic infection. And then specialized transduction is DNA from a specific region of the host chromosome is integrated directly into the virus genome, typically replacing some viral genes. Homologous recombination may occur or may be integrated during lysogeny, and it's a very rare event, but a part of the host DNA incorporated into the phage DNA. All phages are very high efficiency of transducing a cell. It occurs in many bacteria and at least one archaea. An example would be your multi-antibiotic resistant gene in Salmonella, Shiga-like toxins in E. coli, and virulence factors in Vibrio cholera. And that's all I'm going to talk about. And now Maddie's going to go and talk about our other and last topics.
There you go.